everyone. Cotton plants, among many others, are susceptible to a number of insects such as members of subclasses Lepidoptera, Coleoptera and Diptera of Insecta, the biggest class of phylum Arthropoda. Their infective stage is usually the caterpillar stage. These attack the upper part of the plant, badly destroying the lint of the cotton. Protective insecticide sprays to prevent this damage is available but is tedious and time consuming. Bacillus thuringiensis, also referred as BT, is a gram-positive soil bacterium which is known to contain genes encoding insecticidal proteins called delta endotoxins. These are crystalline to look at but are in their inactive form. When the insects ingest this, the crystal reaches their midgut where the high pH solubilizes the BT crystal, thereby activating it to a form that is absorbed by the midgut of the insect. This causes inflammation followed by gut paralysis, culminating in the death of the insect. Considering this property of BT proteins, people thought of utilizing cry protein formulations extracted from a Bacillus thuringiensis culture grown in a bioreactor, mainly to control caterpillars on a variety of crops. These did produce results like their synthetic counterparts but suffered some limitations. Their effectiveness was rather inconsistent due to sensitivity of this free toxin to harsh climate conditions, sometimes inadequate consumption of the toxin by pests failing to kill them, at other times inaccessibility of the pests by the foliar spray, and last but not the least, the older larvae being rather immune to Bt toxin spray. And of course, this kind of exogenous application of the endotoxin was really labor intensive, time consuming and expensive as well. Due to these experiences, RDT experts made a goal of cloning the suitable cry protein genes in various plant species. This was with the great idea of imparting self-sufficiency to them for synthesizing the toxin on their own, thereby preventing their damage by these nasty insects. Most cry proteins are active against specific groups of insects, such as larvae of certain kinds of flies, beetles, and moths. For example, Colorado potato beetle larvae are affected by Cry3A proteins. Cry1AC is used against tobacco budworms, and European corn borers can be killed with Cry1AB, Cry1F, Cry1AC, and Cry9C proteins. Other cry proteins are active against mosquito larvae, flies, and even nematodes. Here, cry stands for the crystalline nature of these proteins. Once the cloning was successful in cotton, scientists wanted to apply this strategy to edible crops too. So, it was important to see how this endotoxin would behave once they get access into the human gut. It was found that these got degraded within just 20 seconds of their entry in the digestive tract of mammals. Hence, it was considered safe to utilize this strategy for creating useful Bt crops to replace the use of synthetic insecticides. This was viewed as not only convenient and hassle-free but also eco-friendly, hoping that now a lesser amount of chemical insecticides would be needed. The GMBT plant-produced endotoxin amazingly overcame most of the limitations posed by the exogenous toxin spray. Hence, this new technology for managing insect pests was approved for commercialization by the US Environmental Protection Agency EPA in 1995. Such GM seeds were distributed to the farmers as well. Now, the genetic part of this worthwhile accomplishment includes three components. First is the cry gene. Earlier, just the Cry1AC was chosen. Later on, a combination of Cry1AC and a gene protecting cotton from application of the common but toxic herbicide glyphosate was preferred. Now, stacked gene complexes of various genes in multiple permutations and combinations are freely available. The second component is the promoter, the crucial one. Depending on the requirement, the promoter selected could allow toxin production throughout the plant or only in some selected susceptible parts and even in a turn-on, turn-off mode. On the safe side, the first type of promoter is preferred if a new construct is being attempted. And the last component is a genetic marker. A selectable marker suiting the host system helps the plant breeders to screen and select the successful recombinant plant varieties. A common marker is a herbicide tolerance gene linked to the Bt gene. Following the cloning, plants are treated with the same herbicide for proper selection. Plants that survive the herbicide treatment are the real successful GM crops. Bt cotton was indeed accepted and proved immensely useful, but reproducing the same approach in plants providing edible products faced a lot of resistance by anti-GM groups. This is attributed to the possibility that despite the fact that crop losses can be definitely controlled by this approach and Bt was appearing to be non-toxic to humans prima facie, yet none can deny that continuous consumption of such plant products for years could cause horizontal gene transfer to human genome anytime and could prove dangerous and irreversible, actually more harmful than beneficial. Guys, worth mentioning is that development of Bt crops has over the years unfortunately given rise to many Bt resistant insects and pests too, and this was actually expected according to evolution principles. How this situation was handled by scientists intelligently will be discussed in some other video. We hope you have liked this simplified narration of the development of Bt cotton. If yes, please write so in the comment box. And do like, subscribe and share this creation. We are grateful to all of you and shall see you again with another interesting video super soon. Keep learning, take care and bye.